can have a high frequency, a higher probability to mutate with respect to other genes. So, at the end, do you think that the environment can direct mutation through the epigenetic modification to a certain gene? And so, do you think that can be increased the rate of mutation, particularly directed to certain genes due to the environment? I don't know if that was clear enough.
Yes, no, that was very clear. Thank you very much for the question. I think the answer is yes. So, under certain circumstances, certainly with the plant systems that we've described, that we and others have worked on, there is evidence that the environment can direct epigenetic changes to certain regions of the genome that are associated with stress responses. So there's evidence there for some direction in the whole process. What we also know from certainly work with animal systems, and there's no reason to think that plants would be different in this regard, is that a C residue with a methyl group on it is more likely to mutate to a T residue than one that does not have a methyl group on it. So I think that what one can think of is, in some respects, as epigenetics, as giving organisms the opportunity to experiment with mutations. The organism can try out within the population. So if there would be selection for epimutations, those would in due course undergo a transition to a genetic mutation. If an epimutation did not have a beneficial effect, remembering, as I referred to in one of my slides, that epigenetic changes can revert, they are unstable, then there would be selection for the loss of the epimutation, and so the population has then gone back to the situation that it had in the first place. So I think it offers, partly, as you say, the potential in some instances for directionality, but it also offers an opportunity for, as I say, experimentation with heritable changes at particular levels.
I understand well the epigenetic mutation can occur in a different environment, in different rates, probably, different intensity. But is there any evidence in particular circumstances of the rate of the epigenetic mutation?
I think we're just, so the question is about whether or not the environment can indeed influence the rate of epigenetic mutation. We're just now using the technologies that I alluded to, just now beginning to generate data about the rate of epigenetic change. And this is studies, for example, with plants, where we can follow them over, in this instance, 30 generations, and where the analyses indicate that the rate of epigenetic change is orders of magnitude more frequent than the rate of genetic change, the rate of spontaneous mutation. I think, as you say, the next interesting analysis to do from this would then be to subject these 30 generation pedigrees to stress at various times or various treatments in order to see whether or not that influenced, as I would expect, the rate of epigenetic change.
Thank you for your presentation. Try to speak English. I don't know if it's good enough. But in order for the stressor to influence next to the future generation, let's say. I'm sorry for a straightforward question, but I don't have the capacity of being so clear. In order for a stressor to influence future generation, they must influence not just somatic cells, but the reproductive cells, because of the separation, of course, of the two lines. Is there some evidence in favor of this instance?
Well, so here you are asking me a question like a molecular biologist, so you need evidence of mechanisms in order to be able to give credibility to, let's say, an explanation for phenomena. In plants, we don't have the separation of the somatic and the reproductive cells. And so it's a lot easier to conceive of mechanisms that can explain transgenerational effects in plants. In mammals, of course, there is the separation, as you say, of the reproductive cells and the somatic cells. So in order for there to be transgenerational effects, what would need to happen is either the stimulus of the transgenerational effect would have to act very early in embryogenesis before the separation of the germline and the somatic cells, or there would have to be the potential for the soma, the somatic cells, to communicate with the germline. In mammalian cells, we don't have an answer to this at present. We do, however, in Coeluroditis elegans, in worms, there's a very beautiful one, showing indeed that there is communication with the germline that is most likely mediated by movement of RNA. So RNA is a mobile molecule that can move between cells. So what we know is that in worms, in lower animals, there is the potential for communication between the somatic cells and the germline through RNA-based mechanisms. For me, I mean, I've worked for a long time on RNA. I just find this very fascinating. And if I worked on mice, I think what I want to be doing is to try and design questions to ask about the potential for RNA to carry out this communication. But you're absolutely right. If these observations, such as the one I described in the Swedish populations, are to have real credibility, we have to be able to reinforce them with an understanding of a mechanism that accounts for communication with the germline.
Thank you very much. Is there any evidence that pollution could produce epigenetic mutation in humans? And are they stable or reversible?
The question is about the effects of pollution.
For instance, air pollution.
Air pollution. Well, I mean, as would have been clear from the discussion, in human populations, talking about evidence for transgenerational epigenetic effects is that, let's say, at present, this is speculation, but speculation that can be backed up with, let's say, evidence from at least other systems that the speculation is at least worth engaging in. I don't think we're in a situation where we can say for sure that any particular effect is any particular transgenerational effect is epigenetic, so pollution or health. But what I would add just parenthetically is that although in this is the evolution day, and so I wanted to focus on transgenerational epigenetic effects. There is a lot more evidence for intragenerational epigenetic effects. So ways that the environment that an organism is exposed to can have long-lived consequences, and that these can be mediated by the sorts of epigenetic mechanisms I am talking about. And, well, specifically in answer to pollution, I don't know. I mean, many pollutants also have the potential to introduce genetic changes. But I think the general rule is that epigenetic changes are more frequent than genetic changes under many different conditions. But we need to get the data to back this up. But I would certainly think that it is something worth looking at.
concerning to evolutionary influence. Epigenetical theory is closer to good punctual equilibrium than to classical neodarwinism, I think. Isn't it?
So, I'm sorry, could you repeat the question?
Is epigenetical view is closer to good view of evolution than classical neodarwinism, or not?
Well, I think, I mean, epigenetic understanding can be, it could be reconciled even with, the original Darwinian view of evolution. But I think that several of the questions that have come up this evening have been about the quantitative aspects of the epigenetic effects. So what is, I think the question is, the important question is, what is the relative contribution of epigenetics as opposed to genetics? I mean, so genetics, of course, is the framework. It underpins absolutely everything about living organisms. We can't deny the importance of genetics. But then we can think about epigenetics as being superimposed on genetics. And I think where we are at present is, as I tried to convey this evening, we have enough information from some organisms to know that yes, epigenetics most likely does play a role in evolutionary transitions. What we can't say at present is, to what extent they do this. And I think that before we can really assimilate epigenetics into, let's say, the theorizing about evolution, we just need this information about, the quantitative information, the relative extent to which epigenetic changes influence heritable phenotypes. Would you not agree, or?
Rapid changes or gradual changes in epigenetics? You prefer rapid changes or gradual changes in organism evolution?
I think it's the heritable changes that would be the ones of particular interest in an evolutionary context.
Is there any experimental evidence that the epigenetic change affect in a different way nuclear or mitochondrial or chloroplast DNA?
The sorts of epigenetic mechanisms that I'm talking about are predominantly with nuclear DNA, and I'm not aware of any evidence that could really incorporate or reconcile this view of epigenetics with mitochondrial or chloroplast DNA.
May I ask you something? This evening, you don't have touched the point which in my mind as a kind of evolution is very important, and that is how far the epigenetic changes may be adapted. I want to accept that epigenetics is a reality, but my interest is on epigenetics as a mechanism, a real mechanism of evolution, not of laboratory experiment of evolution. So the question is, for example, it comes to my mind the example of Inagria. I don't see any good reason to believe that Peloria is an adaptive phenotype with respect to Inagria. They may have exactly the same fitness value. Or in the observation of this Swedish population, how is this change adaptive?
I think one could think of this in two ways. So, in one way of thinking about it, the epigenetic change, let's say, would be so much more rapid than genetic change that there is an adaptive value to that. So if you've got an organism, let's say, which is subject to, let's say, an evolutionary transition. For example, it's a plant growing on a planet that is getting to be about 4 degrees hotter than it used to be. And if the temperature stress induced very frequent epigenetic changes, even though those epigenetic changes would not be directed in any way, one could conceive of so the possibility that this rapid, the high frequency epigenetic change generates heritable variation that could then be a substrate for natural selection in a new environment in which the plant finds itself. So one can think of it in those terms. But there are data from plant systems in particular, well, this is what I know best, where plants have been subjected to disease stress, for example, and where it turns out that in subsequent generations, the defense systems of the progeny are primed such that those plants are more resistant against the disease agents. Now it may look so, that is a system that has direction to it. It may well be that there is and there almost certainly will be a cost to that type of heritable epigenetic change. But then it would be interesting to see, let's say, depending on the infection pressure by the disease agent, whether the cost outweighs the benefits of the heritable resistance to the disease. So I think there are examples, certainly with plants, of how the heritable epigenetic change could be directly impacted. And I think, as I say, just to recapitulate, one could also conceive of scenarios in which, even if there was no directionality to the changes, the speed could confer an advantage to the population that underwent those changes.
In your model, from microRNA, you go and get methylation of the coding sequence. Okay? How do you explain methylation in the promoter region?
So this is how can it be that RNA can affect methylation changes in DNA and in the promoter of genes? Again, this is molecular biologist arcane stuff, but what we now know is that essentially all of the genome of organisms is copied into RNA. And that parts of the DNA have RNA attached to them. And it's this RNA which is the target of the small RNA that mediates the epigenetic change. So the epigenetic changes are actually mediated by RNA-RNA interactions rather than RNA-DNA interactions.
So you need the distance scribe also the promoter.
I'm sorry?
So you need, but also the promoter should be transcribed.
Well, indeed, and as I say, there is now good evidence in many organisms, humans included, that most of the genome, including many promoter regions, is indeed transcribed into RNA at a very low frequency, but nevertheless it is transcribed.
May I ask you what triggers the DNA methylation? Sorry.
Yes.
What does the methylation, the DNA methylation trigger?
So what is the trigger of DNA methylation? I think that there are probably several answers to that question. But one of the answers would be that external stimuli trigger the production of the RNA molecules, which I illustrated to you, which can then mediate the introduction of the methyl groups into the DNA. So these would be the factors that can be influenced by the environment. And then of course, the question is, well, what triggers the activation of those factors. I don't know.